Hello, and first and foremost, thank you to the organizers for the invitation. Hang on, sorry. Okay, perfect. All right, so I'm going to talk to you about CAR T cells and CAR T cell therapy and CRISPR. Currently, I'm a postdoc working in Professor Kasim's lab. So we're based at Great Ormond Street Hospital, but at the Institute of Child Health, which is fused to this building that you can see up here. If you don't know where it is, it's close to Russell Square in London. It's one of the largest pediatric hospitals in the UK and very well renowned. They carry out over 100 stem cell transplants per year from different sources and for different conditions, and quite a lot of rare metabolic disorders, rare immunodeficiencies. And there's over 2,000 patients treated every year. So I work in the molecular and cellular immunology unit, and what we've focused around, so this framework basically shows you from 2010, the different PIs in the section have brought forward all these experimental treatments. So you can see there's quite a few HSC treatments, but the ones that I focused on, and Professor Kasim, is T-cell therapies and skin-related therapies. A lot of these have now progressed past phase 1 into phase 2, and I will be talking. Oh yeah, sorry. So these treatments have relied on two separate strategies. Predominantly, it's been gene addition. So we've not heard a lot about it today, but we rely on retroviral, lentiviral vectors to shuttle a therapeutic transgene within a cell of interest. We've now moved to the safer lentiviral constructs, stripped of all virulence factors, so they're replication deficient. And we can package up to 9 kilobases of genetic material, the cDNA, that could be used to restore protein expression. An example of the largest thing we've put in was for collagen for the treatment of epidermolysis bullosa, which has now gone into phase 1. More recently, however, we've also joined the field of genome editing, and this sort of kicked off in 2010 with zinc fingers, moving on to meganucleases, TALENs, and more recently CRISPR. So we've gone through all those steps as well, and trying to incorporate it into our current treatments. I'm sure you've probably all heard about this, but with the gene editing, you could have one of two outcomes. You could either have an ablation of gene function to remove allo-reactive epitopes, for example, or you could go down the homologous directed repair pathway, where you could introduce a template of interest. This could be just a short SNP change, or it could be a full-length cDNA, and you could do this in a non-viral approach. So this is the general pipeline, obviously, that everyone that works in a translational field will follow. You've got the R&D stage, the translational stage, and then moving into phase 1, 2, 3, and then licensing the product. So we operate within a very small part of this, really. The first few years, which academia would only go up to phase 1 and 2, and then industry would take over for the licensing. And we particularly operate within this section, and I'm just in the first 2 here. Then it gets taken over by the GMP personnel. So the focus of this talk will be on B-ALL, acute lymphoblastic leukemia, and looking in the pediatric setting. So this is the outline, but also what's been developed for this treatment, for this disease at the moment. So we have standard care, which is the bone marrow transplant if there's a suitable donor. Recently there's been TALEN edited universal T cells. There's obviously autologous therapies, which we'll go into as well. We've now progressed this to a CRISPR edited universal T cells. I'll show you about the assessment of on and off targets, which is one of the primary considerations with using gene editing tools. And I'll just touch on CRISPR uncut, which is the new kid on the block with the base editors. Okay. So we've not been introduced to CAR T cells yet. I know Dr. Silk mentioned the transgenic TCRs. So this is a progression from transgenic TCRs. They're not necessarily better, but what is predominantly different about CAR T cells is that it's the chimeric antigen receptor. So it's a fused short chain variable fragment of a monoclonal antibody onto the T cell receptor, onto components of the T cell receptor. So what you would typically have is your scFv. This has been mostly, again, CD19 because it's ubiquitously expressed on B cells. It's not specific for tumor cells, so you would get total ablation of them. This is fused via a linker in a CD8 transmembrane domain to a co-stimulatory domain. So there's been various iterations of CAR T cells, and they've gone through a number of generations over the past years. And they have worked out that adding this co-stimulatory domain is critical in the function. So this could be either a 4-1BB or CD28. They have various effect on persistence and potency. And the last portion would be a CD3 zeta chain for transferring the signal. So the talk that I'll give you is based on second generation CAR T cells. So this is the standard scFv with one co-stimulator domain and the CD3 zeta chain. There's now generations with double co-stimulator domains. And just a key difference to transgenic TCR and a CAR T cell is that the CAR T cell is not HLA restricted. And that's one of the key differences. Also, with switching out the scFv, you can have a different function targeting a different antigen. We have a number of scFvs because there's a number of clinically approved monoclonal antibodies, so it's fairly straightforward to swap these around. You would have heard about CAR T cells, most likely in clinical trials at the moment, because we've now had the release of two clinical products. So Kymria and Yescarta. So this was development from University of Pennsylvania, from Carl June, and this was a report on it. So you can see that the survival rate was about 50% after 12 months, so it's a moderate improvement. These were adult patients, and predominantly they're refractory relapsed. So they've tried bone marrow transplants, they've tried chemotherapy. This was a last resort treatment. There were levels of GVHD detected in the majority of patients, and these were treated with drugs such as Tocilizumab. It's an IL-6 antagonist. Nevertheless, these have made it into a therapeutic product and are available from Novartis or Kite Therapeutics. So these two treatments, they are autologous treatments, and what this is, it's the next slide. But the fact that they're autologous treatments means that you have to use the patient's own cells. There are a couple of limitations, especially when you're looking into the pediatric field. So you're talking about very sick patients that have undergone a number of treatments. Trying to get a usable starting material from these patients is quite difficult, and it will be very variable. Also, it will end up being a bespoke product for each of these patients, making it extremely costly. And this has been even more so the case in the pediatric field. Additionally, there's been a couple of reports where targeting the CD19 has led to CD19 epitope escape through mutations in the exons, causing it to be downregulated, and the CAR T cells can't detect it in time to kill it. This could be the case when trying to target it with any CAR T cell, but the more potent it is to begin with, the less chance of it escaping through these mutations. So this schematic is what I want to show you. So the autologous approach involves taking cells from the patient, then modifying these ex vivo using a viral vector of some sort, carrying the therapeutic, the targeting CAR. So it would be carrying CAR19 within here. The T cell that you will produce will still have all these receptors on, and once you've made enough of it, you can supply it back in, reinfuse it back into the patient. This is potentially, the cells could potentially be permanently within the patient. It could cause some form of B-cell aplasia, but typically the patients would undergo a further transplant. What we focus on and what our group is all about is the design of universal CAR T cells. So the difference is that we would start off with a healthy donor. We would get a leukapheresis, which we would then transduce with a viral vector, but also manipulate with gene editing tools to remove cell surface receptors that would make it be recognized as foreign in the recipient. This has the CRISPR molecule here, but it could be TALENs, it could be zinc fingers. Once you've knocked out the receptors, you're expressing your CAR. Again, you undergo the same process of ex vivo expansion. But then what you can do is cryopreserve doses and give this to a number of patients as an off-the-shelf treatment when required. This is precisely what was done in the Selectis now Servier trial, which was piloted originally in ICH. So they used the previous gene editing tools, so TALENs. So they took the single donor material, activated, transduced with a CAR19 transgene, knocked out TCR and CD52. So CD52 is a target for Alemtuzumab or Campath. This is a chemotherapeutic drug. So it's a way of protecting the cells from being killed off by Campath, which has a very long half-life. Following purification, you obtain a fairly pure yield. It will be TCR negative, but it won't necessarily be CAR19 positive, and that's a key thing to remember and what differentiates our next trial from this. And then you'll design this off-the-shelf product. This is the way TALENs work. The only reason I left it in is just to show you that they're fairly large molecules. In this case, they were delivered by mRNA, so you have two pairs of mRNA that you have to electroporate into the cells. They're expensive to produce. They can't be delivered through a viral vector because of the complexity of the sequence, so they have certain limitations. Nevertheless, they work very efficiently. So these are the plots of what you'd expect throughout the production of these cells. So your T cells, when transduced, would have a certain transduction efficiency. This is particularly high. It could be lower. It could be 40%. And again, this is a key factor for future trials. Following your TALEN-mediated knockout of the targets, what you'd expect is a TCR and CD52 knockout. This plot here is actually showing you the depleted population. So you would originally have had 4 populations following that TCR and 52 knockout. And this population up here, you don't have the percentage here, but it's 0.7. This is the residual amount of TCR positive cells, and this is what could kill a patient if the thresholds aren't kept to a very strict limit. So we have a limit of 5 times 10 to the 4 cells per kilo, which can be TCR positive. If it's beyond that, you would cause severe GvHD in the patients because of the foreign TCR. The CD52 knockout will self-purify because of the CAM path within the patient. And this was the study that described 4 of the patients. There's now been quite a few more treated with this therapeutic, again from the same first donor batch of cells that was produced. This was just to show the patient, the marrow in the patient. So after, these were relapsed refractory BALL pediatric patients that had failed, again, all other treatments. And this was a last resort treatment for them. But upon delivering the CAR T cells, they went into MRD negative status. This happened within the first 28 days. After that, they received conditioning to clear the residual CAR T cells, and then they received an allogeneic bone marrow transplant. It's key to remember that what we're attempting is a bridge to transplant therapy. So we start off with trying to eliminate the leukemia, then delete the host immunity using anti-CD52, the lymphodepletion, and then the patients will receive a matched transplant. So these cells, the CAR T cells that we want to make, won't persist. And this was the famous case of Layla, which was one of the plots that I showed you earlier. So we know that this does work, and she's now healthy and going to school. So she was treated when she was about 9 months old. Oops. Okay. So moving on to CRISPR now, which is what I've mainly focused on. This is just to show you that there's a number of delivery methods now open to us because of the smaller size of CRISPR. It's less complex. It can be split into its different components. You could deliver it with a viral vector as naked plasmid DNA. You could split your short guide RNA sequence, which detects the sequence, and the Cas9, and you could deliver those as RNA and mRNA through electroporation, or you could couple them and create an RNP, a ribonuclear protein, and electroporate that into cells. So it's very versatile. And this is what I wanted to show you earlier. So up here you can see what the TALEN CAR treatment would look like. So you'd end up with these 4 populations, and even once you've done the TCR alpha beta depletion, you will end up with CAR negative, TCR negative cells. These won't have much use in the patient, but they'll still be in your dose, which means that your dose is not homogeneous and is reliant on the initial transduction. So if that's low, then this will outweigh the actual effector dose. You can't push it up because you're capped by that TCR positive threshold. So we thought if we put part of the CRISPR component within the viral vector, we can couple this effect. So only cells that have CAR could have the option to be knocked out. So we eliminate this. So after purification, we should get something which is CAR positive, TCR negative, and eliminate that population. So we did quite a lot of vector design, and to do this we designed a terminal vector. That's what we've called it because we've cloned the sgRNA portion of the CRISPR into the LTR, the long terminal repeat. When it goes into the proviral stage, it self-duplicates into both LTRs, and importantly it doesn't interfere with expression of your transgene. That means when we infect, when we transduce the T cell, we'll have sgRNA expression, and then we'll transiently electroporate Cas9 because we don't want that hanging around, and we'll end up with a cell which has CAR and is knocked out for TCR. We've also applied this to CAR20, CAR22, and CAR123, amongst others, for the targeting of various disease. Okay. So this was just to show the duplication actually occurs. So when you put it in one LTR, it will self-duplicate into the other one, increasing expression. And we optimized the day of electroporation with a Cas9 based on when the highest expression of the sgRNA was. And we can see the transience of the Cas9, which is diluted out in culture after about 48 hours. At the functional level, at the protein level, we can see that this is titration again, but when you give an increased amount of Cas, you get knockout, which is restricted to the CAR-positive population. And using TIDE, which is based on Sanger sequencing and deconvolution of the sequence traces, you can see the indel pattern. So we scaled this up on a semi-automated GMP-ready machine, Clinimax Prodigy. It's a closed system. We start off with 100 million cells, and following transduction, electroporation, and TCR beta depletion, we could get a very, very pure CAR-positive, TCR-negative population, which was 3 billion starting from the original 100 million. So this could be enough to treat 20 patients using that same batch, which would be the whole of a phase 1 study. We tested this in vivo, of course, against a conventional CAR19 vector. This is without the knockout. And what we saw was there was clearance of the Dowdy tumor with luciferase. This was non-invasive bioluminescence screening for tumor progression. And then looking in the marrow, we could see that tumor had cleared, and the longer we left it, the clearance was more significant. Whereas in the conventional setting, without the knockout, there appeared to be re-emergence of tumor. We looked a bit further into this, and we noticed that even in this Dowdy, which is a cell line, we saw what looked like the CD19 escape that was reported even in the clinical setting. And you get CD19 negative but CD20 positive. This wasn't the case with our terminal construct. When you look into the effector, so the T cells, what we noticed was there was a huge upregulation of PD-1, which is a known exhaustion marker. And this is probably because of the TCR still being expressed on the cells. So we believe it might have caused the cells to be slightly anergic and couldn't clear the tumor by that stage, whereas the terminal cells were still PD-1 negative. We've obviously tried this now with TTCAR20, and we've performed the same experiments both in vitro and in vivo with 51 chromium assay. So it's a cytotox assay, in vitro assay, and also in vivo studies, and it's performed as well as, and perhaps even better than, CAR19. So we do have this in the pipeline as a backup to 19 if we see this escape, or it could be used from the beginning as a combination. TTCAR123 is another option for AML, perhaps. What we've also managed to do is multiplex the effect. So that original Selectis trial, as you heard, knocked out both TCR and CD52. So we've managed to introduce a second guide within this construct. We had to manipulate the promoters and make minimal promoters. But following a double depletion, we can get, again, a very pure population. We've tried this with B2M to knock out HLA class 1 and TCR. And we've also triplexed this by keeping the two within the construct and then also delivering a separate sgRNA. So we can also get a TCR negative, class 1 negative, 52 negative, CAR positive population by the end of it. So there's a lot of options with this system. Obviously, the more hits you have on the genome, the more chance of having some deleterious effect. So we have to very carefully screen this product at the end. We've got multiple ways of doing this. We start off with karyotyping and FISH analysis, Sanger sequencing and targeted NGS of in silico predicted sites and actual sites. In silico predicted translocations, we also look at this as a combination of NGS and karyotyping. Actual off-target sites, I'll show you this. There's assays now developed by other labs that can assess in vitro cleavage, and it's not purely in silico. Whole genome sequencing is fairly reliable, but it has to be coupled with all these to be meaningful. So this was from the original TALEN trial, but there were a number of in silico predicted sites, so you can plug your sequence within some software, online freeware, and it will try and detect potential off-target sites based on 1 to 5 mismatches within that guide sequence. So once we have these, we can then run a targeted NGS on them, as well as an on-target NGS on them. This is the FISH analysis, so we've got probes on the TRAD locus which flank the track cutting site, the TCR cutting site, and you can see based on the colors is red and green, so if you see one and not the other, it means that there's possibly some translocation, which is known to happen on chromosome 14 regardless of the cut. But there were 4% translocation events detected within those Selectus products, which we don't know what their effect may be in vivo. They might be deleterious to the point where they're eliminated, but it could potentially be harmful. That we don't know yet. These are all the potential translocations once you introduce 2 breaks in the genome. And karyotyping, you see on that product, you had portion of chromosome 14 go up here to chromosome 1, which is where the CD52 knockout occurs. So it does happen. It's not just our imagination, it does actually happen. We also perform, this is NGS results, so we can detect the indel pattern and whether it's substitutions, deletions, insertions. And this is the true in vitro cleavage. So in vitro cleavage, as opposed to the in silico, is where you digest your untransduced DNA, just normal DNA, with your guide of interest in the Cas, and you'll get a pattern. So this will give you a lot more information as to whether a site may be potentially hit by your specific guide. We did this on our sample that came off the Prodigy, and it flagged up quite a lot of sites. But then when we actually do NGS on the actual treated sample, we saw that none of these came up. And lastly, we did whole genome sequencing, but this is only 30x, so it will just tell you that there's not much difference, no significant variance between the untransduced sample and the knocked out sample. But we can't really take this any further because of the resolution. There are multiple artifacts that you could interpret completely wrong if you don't have other systems to back it up. And this is now going into trial in June. We've patented the vector construct, and we're preparing the therapeutic IMPD for use from June. And just in the last couple of minutes, I just want to tell you that we have a new advancement. So this is base editing technology, and it's a result of this and trying to improve on this. So there were 2 publications that came out very recently from the Liu Lab, where they fused APABEC, which is typically used to convert mRNA CTU, but they fused it to a catalytically inactive Cas9. That allows C to T conversions, which can be very beneficial to us without causing a cut. So we've used it alongside, so this is the standard Cas9 mRNA that I showed you previously, where we get the knockout, and now we've done it with a codon optimized base editor. Again, we get true protein knockout, but at the genomic level, there's no cut. And what we've managed to do is find tryptophans within the sequence that would create a stop sequence, and instead we'd stop the transcription rather than actually cause a break. And this is a double knockout without genomic cleavage. So you can see how you could have multiple knockouts and reducing the potential of these deleterious effects from the genome breaks. Here we targeted the splice donor site for CD52. And you can see from this gel that when trying to look for the translocations with the SP-Cas9, the standard Cas9, you get all these translocation effects that you would have seen with TALENs as well, but with a B, you don't. These are just the endogenous sites that come up for 52 and TRAC. And yeah, the last slide was that we've now managed to triplex this as well, so we have a triple knockout of TCR52 B2M with base editing technology. Obviously, this can be used for SNP modification. Most of the pathogenic mutations are A to T, G to C. So now there's base editors that could convert those. So it's not its use could be quite broadly expanded to such other diseases. So just to finish, we talked about universal T cells and how we think this is the way forward for CAR T cell therapy. We're now moving into phase 1 trial for this current vector, but hopefully we'll be able to knock out enough of the other HLA restriction factors to broaden the use of this vector. And lastly, just I hope you appreciate how fast this technology is moving, that we can't even keep up with it. So in a week's time there'll be a new base editor out, but we're testing all the potential options to have the best outcome in the patients. And I'd just like to thank obviously Professor Kasim and Roland, who did most of the work on this, who's a PhD student, all our funders, and thank you for listening.